This is a critique of 3 figures from a 2011 Journal of Neuroscience paper, and it includes analysis of the original data. I'll refer to myself as graduate student and to the authors as first author, second author, third author, because I want the focus here to remain on the science and not on the people involved. I should preface all this by saying that this is my opinion and to the best of my understanding, but it's an informed opinion as I was a graduate student in this lab for over 2 years and have since analyzed the original data. It's my opinion that these figures do not accurately represent the data or the experimental paradigm. And further, I think that the conclusions of this paper are not supported because the analysis throughout this paper really just consisted of comparing one sample of a layer 2-3 sink with a second sample of layer 2-3 sink. So throughout, they were comparing layer 2-3 with itself. So I've analyzed the original data from figures 1A, 1B, 3A, 3B, and figure 5A. And I apologize in advance for the length of this presentation, but it's just going to take me a while to get through all of that data analysis. Now, there's a few things that all 3 of these figures have in common. First of all, there is no quantitative y-axis. By that I mean that there is no cortical depth in microns on the y-axis right here. Instead, there's this categorical y-axis with an assignment of laminar boundaries. And another thing that these have in common is that layer 1 of cortex is placed above the surface of the brain. Now, these recordings are made from the full depth of the auditory cortex of a mouse, and yet layer 1, if you look carefully on here, is actually placed above the surface of these recordings, so it's really being placed above the surface of the brain. So it doesn't really make a whole lot of sense for layer 1 of cortex to be above the surface of the brain. And it's really noticeable here in figure 3, where they just don't even put layer 1 on the categorical y-axis. Now, I'm going to analyze the data from figures 1A, 1B, 3A, 3B, and 5A. And before I really get into everything, I want to take a quick tour of figures 1A and 3A and 3B. So this is my outline. I'm going to give you that quick tour of figures 1 and 3. Then I'm going to give you everything you need to understand the background and the experimental paradigm. Then I'm going to give you a full quantitative analysis of the original data from figures 1, 3, and 5. Then I'm going to talk about my own experiences in graduate school, trying to interpret these multi-probe recordings with a quantitative y-axis. And finally, I'm going to close with some questions and concerns that I have regarding this paper. So let's start with figure 1A. Here's the original figure 1A. Now, first of all, notice that there is a categorical y-axis right here. These are traces from the auditory cortex of a mouse, and they used a 16-channel multi-probe. So there's 16 channels here, and yet actually there's only 13 channels shown. So there's 3 channels missing. And there should be 14 CSD traces, but there's only 11 CSD traces being shown. And readers, editors, reviewers are given no idea of what cortical depth these traces are recorded from. There's no quantitative y-axis, and there's also no y-axis scale bar. Now, what I'm going to do throughout this entire presentation is I'm going to use a figure from a paper from the same lab from 2007. So this is the lab's 2007 Nature Neuroscience paper by the exact same authors. And what's interesting is that in this 2007 paper, in which they used the exact same paradigm, this is multi-probe recordings from the auditory cortex of a mouse, using the same 16-channel multi-probe. In this earlier figure, they took a very straightforward and very quantitative approach to representing the data and to communicating the experimental paradigm. So here are the 16 channels of the multi-probe. There's 16 LFPs. So these are recorded throughout the depth of auditory cortex, and auditory cortex in mouse is a little over 1 millimeter in depth. So a millimeter has 1,000 microns in it. So the depth of cortex, the full depth of cortex would be down to about right here. Now, what's done is something called current source density analysis, and that gives you a picture of the spatial temporal patterns of activity in the brain. So this y-axis is time, this x-axis right here is time going left to right, and this is cortical depth on the y-axis. And what you see is that in response to sensory input arrives in layer 4 in the middle layers, and then activity spreads up to layers 2 and 3. Now, layer 2-3 is at a depth of 200 to 300 microns in mouse cortex. And what you see here is that what the authors did, it was just compare two traces in the same superficial sink in layer 2-3. So their entire analysis for several years really just consisted of comparing two samples of the same superficial sink in layer 2-3. They were just comparing layer 2-3 with itself. So this is really a rather embarrassing thing. It's embarrassing that they spent 7 years comparing layer 2-3 with itself. And it seems to me that this figure really tries to obfuscate that fact. And if you look here, this is entirely analogous. They're analyzing this red trace and this green trace. Really, everything is the same. They're making the same analysis. They're just not being forthcoming with editors, reviewers, and readers about what depth these are all recorded from. And notice that they slyly put layer 4 right here. But layer 4 is just, it's not at a depth of 300 microns. It's deeper, and I'm going to show you that later on when I show you some of the anatomy. Now, this figure it's lacking a categorical y-axis. I'm sorry, it has a categorical y-axis. It's lacking a quantitative y-axis. It lacks a y-axis scale bar. 3 of the LFP traces are missing, and 3 of the CSD traces are missing. So I have access to the original data, so I'm going to analyze some of that and put that right here. So here's my analysis of the original data, and what I have here is grid lines for each one of the CSD traces. So there are 14 CSD traces. And I'm going to add in a quantitative y-axis. Well, first I'm going to add in the missing LFP and CSD traces, and then I'm going to add in the quantitative y-axis. And you can see that the entire analysis throughout the paper consisted of comparing a trace recorded at layer 2 at a depth of 200 microns with a trace recorded at a depth of 300 microns. So all they were doing was comparing layer 2-3 activity with layer 2-3 activity, which is really rather embarrassing and tragicomic. So a depth of 200-300 microns is solidly in layer 2-3. In fact, if you look at this sink is actually at a depth of only 200 microns. And this is just a figure from the Allen mouse brain atlas showing that a depth of 200 to 300 microns is solidly in layer 2-3. Now that was figure 1A. Now I'm going to show you figure 3A and 3B, which is really quite an astounding figure because it's missing layer 1. Now here's the original figure. Again, notice that there is no quantitative y-axis. There's no y-axis scale bar to give readers, reviewers, and editors an idea of what cortical depths these were recorded from. And again, these recordings are from the full depth of auditory cortex. So why is layer 1 of cortex not on here? So I analyzed the original data. Here it is on the right. Here, each one of these grid lines is one of the 14 CSD channels. And this is the full depth of auditory cortex. So this is really a very superficial sink at a depth of 100 microns, which means this is a layer 1 sink. And a depth of 200 microns is in layer 2-3. Now the other problem with this is that notice right here, layer 4 is at a depth of 200 microns. And they put the entire depth of layer 4 at a depth of only 200 to 300 microns. But that's simply not so. Layer 4 is at a depth of 400 to 500 microns. And of course, the real question is why is layer 1 above the surface of cortex? And this is really I guess at best you could say that this is a failure to apply Occam's razor. What's the simplest explanation of something? The simplest explanation of something is that, well, you recorded this sink at a depth of 100 microns, so it's a layer 1 sink. It's a superficial layer 1 sink. That's a much more straightforward explanation than positing that layer 1 is above the surface of the brain, which is a pretty wild hypothesis. So here's showing that even 300, which they mark as their lower border of layer 4, is still in layer 2-3. So their conclusions are not supported. And layer 4 doesn't even start until you get down to 400 microns. So them saying that layer 4 starts at 200 microns is just unfounded. Now I can do the same thing with figure 3B. In fact, in this case, the analyzed sink was at a depth of only 100 microns, which puts it up in layer 1, or at the border between layer 1 and layer 2. And again, why is layer 1 above the surface of cortex when these recordings were from the full depth of auditory cortex, as shown here with the analysis, a quantitative analysis of the original data? So now I can give you the background and experimental paradigm. So this is a figure from a paper from Charles Schroeder's lab. This is the experimental paradigm as is done in macaque primary auditory cortex with a 24-channel multiprobe. And what you see is that the multiprobe is inserted across all six layers of auditory cortex. And they do something called current source density analysis. You don't really have to understand the math of the computation behind that. You just need to understand that current source density analysis will give you a picture of the spatial temporal patterns of activity in the brain. So left to right here on the x-axis is time. And on the y-axis here is cortical depth. And the patterns of activation that you see is that in response to sensory input, there's this pattern of activity that starts in the middle layers and then spreads up to layer 2, 3. And this is really the canonical microcircuit of cortex, that input comes into the middle layers and then goes up to layer 2, 3. Now on the right here is actually one of my own recordings, my own multiprobe recordings in auditory cortex using a 16-channel multiprobe in mouse auditory cortex. And I really saw a very analogous pattern of activation where I saw initial activity in the middle layers at a depth of 500 microns, followed by more superficial activity up in layer 2, 3. Notice this is a depth of 200 microns, a depth of 100 microns. Now, unfortunately, I was never able to present this at posters or publish this or share it with anyone because this was completely at odds with my lab's framework. So this is my lab's framework, and this is from a very recently published Journal of Neurophysiology paper which just came out a couple months ago. And notice this tendency in the lab. Now, in that picture, that figure that I showed you earlier from 2007, there was a quantitative approach to presenting the data, and there was a quantitative y-axis. But the lab has recently adopted this trend of avoiding a quantitative y-axis, and instead they have this categorical y-axis where they have an assignment of layers. And they clearly peg both layer 4 and layer 2, 3 on this very superficial sink, which is really at a depth of only 100 to 200 microns, which is simply not layer 4. This is a very superficial sink, and that's not founded. And they peg layer 6 on this early sink, which just happens to be in the middle layers at a depth of 500 microns. Now here is a coronal slice showing auditory cortex over here, where it says AUD-P, that's primary auditory cortex. And you can see here with the scale bar from the Allen Mouse Brain Atlas that primary auditory cortex is about a little over a millimeter in depth. This is 1100 microns right here. Now here's the multiprobe that we insert into primary auditory cortex. And here's a little inset showing you the 6 layers of auditory cortex, and that layer 4 starts at a depth of 400 microns. Here's another illustration just showing the positioning of the multiprobe. We have these 16 recording sites. The very top recording site we always had above the surface of cortex. And here are the 14 CSD channels. Now there's 16 recording sites and 14 CSD channels because you need three recording sites to give you one CSD channel. So they're recording local field potentials at each of these 16 recording sites. And you need the information from 3 local field potential recording sites to make one CSD channel. So that's why we always had one site above the surface and the second site right at the surface. Now what I've done here is I've pasted in the anatomy from a paper by Anderson and Linden. This is Anatomy of Primary Auditory Cortex, which we know is about 1,000 to 1,100 microns. So just looking at this a priori, before even doing any experiments, we will know that we'd expect to see activity in layer 4 in the middle layers, right here at a depth of 500, and that activity in layer 2, 3 is going to be up here at a depth of 200 or 300 microns. Now here's an earlier figure from Willard and Ryugo, again showing that thalamic input mainly arrives in layer 4. Now these dots are supposed to be thalamic input. And again, looking at the Allen Mouse Brain Atlas, we know that layer 2, 3 is at a depth of 200 to 300 microns. Layer 4 starts at a depth of 400 microns and extends down to 500 microns. Now here is the quantitative approach to representing the data and the experimental paradigm as was shown in the 2007 Nature Neuroscience paper. Again, notice that the analysis in the lab consists of comparing a trace at a depth of 200 microns with a trace at a depth of 300 microns. So their entire analysis throughout this paper, and really all of the analysis in the lab, has just consisted of comparing layer 2-3 with itself, which is really a meaningless analysis. Now here's a comparison with the anatomy, so that you can see that this sink right here is really the early sink in the middle layers, and this is a sink in layer 2-3. In fact, it's probably a sink in layer 2, upper layer 3. Now, the really sad thing, really, I guess it's tragicomic or it's really just a missed opportunity, is the fact that the lab never analyzed this early sink right here. It was really just completely ignored. This was excused as being a layer 6 axon collateral, but layer 6 is all the way down here. So it's pretty hard to call this a layer 6 axon collateral when it's about 500 microns away from layer 6, and it just happens to be right in the middle layers. It just happens to be right where layer 4 is. So it's really a mistake and a missed opportunity that the lab never analyzed this middle sink, and instead they just compared two samples of the same very superficial sink in layer 2-3. Here's again using the anatomy from Anderson and Linden, just so that you can see that this is solidly up in layer 2 really, and this is most likely layer 4 input in the middle layers. And again, just to show you the explicit framework in the lab, even as recently as a couple months ago in 2012, you can see that they clearly call this early sink in the middle layers, they call that a layer 6 axon collateral, and they peg both layer 4 and layer 2-3 up here. In fact, this really tells you what their framework is. Their framework is that this is layer 4 activity and this is layer 2-3 activity. And that by comparing these two traces, they're comparing early layer 4 input with later layer 2-3 activity, which is just not founded, it's not supported by a quantitative approach to representing the data. And again, this is a comparison of the lab's framework on the left with my own multi-probe recordings on the right. And this is also a shame from my own personal perspective because I was never able to present my data at SFN or at ARO or to publish papers because to present my data in a straightforward and quantitative way was contradictory to my lab's established framework. So I was met with a lot of resistance. And that's to put it lightly. That's the most polite way I can think of saying it. So analysis of the original data from figures 1, 3, and 5. So I'll start with figure 1a. So here is the original figure. Again, notice that there is no quantitative y-axis. There's just a categorical y-axis. Readers, reviewers, and editors are not given any idea of what cortical depth these are recorded from. There's also no y-axis scale bar. And there are 3 LFP traces missing and 3 CSD traces missing. Now here's my analysis of the original data. Again, each one of these grid lines is a CSD trace. Here are the missing LFP and CSD traces. And here is a quantitative y-axis so that we can see that these analyzed traces were at depths of only 200 and 300 microns. Their entire analysis consisted of comparing two traces in the same layer 2-3 sink, thus effectively comparing layer 2-3 with layer 2-3. And for all of this, my template is the straightforward approach to representing the data as was shown in the 2007 paper that used this exact same experimental paradigm in mouse primary auditory cortex. So it's very clear comparing these figures that the top trace is above the cortex, the second trace is right at the surface of cortex, which of course begs the question as to why they put layer 1 above the surface of the brain right here. How can layer 1 of cortex be above the surface of the brain? And again, this is going to be sort of a mantra through all of this. All they did was compare a trace at a depth of 200 microns with a trace at a depth of 300 microns, which is layer 2-3. All this is layer 2-3. And in fact, the sink is really centered at a depth of only 200 microns. It's really a layer 2 sink. Again, just showing you the anatomy again from the Allen Mouse Brain Atlas that a depth of 200 and 300 microns is in layer 2-3. Now, what is shown right here is where I have the same data that is showed you in the previous slide, but everything is matched up in both the x and the y axes. So in this figure, notice that this scale bar here, they do have a scale bar for the x-axis. So this is 50 milliseconds. So left to right in this entire color plot is 100 milliseconds. But if you look here, the x-axis is 0 to 50 milliseconds. So what I do on the next slide is I just analyze this original data, but I bring the scale bar in alignment with this. And that's what you see right here. So this is everything, both the x and the y-axis, completely in alignment. And you can see just the complete parallel here, how this experiment was clearly done exactly the same way, because you see the same pattern of activation. You see this blue source up here, and you see this red sink. And you can see that this sink is just solidly in layer 2-3 at a very superficial depth of 200 microns. So that was figure A, 1A. Now I can do the same analysis with the original data from figure 1B. Now here's the original figure 1B. Notice that there is no categorical y-axis. Here I'm going to add the analysis of the original data. Here I've added a quantitative y-axis, and I've also added in the missing 3 LFP and CSD traces. And here is the anatomy of auditory cortex, so that you can see that this very superficial sink is at a depth of 200 to 300 microns and is in layer 2-3. Now this red trace right here is supposed to be layer 4 activity. In fact, if I go back here a little bit, see if you look at this here, when they draw this line right here, they're trying to say that this is layer 4, that this is their measure of layer 4 thalamocortical input. But that's simply not supported by a quantitative approach to analyzing the data, where we can see that this is just layer 2-3. They're missed layer 4 entirely, and this is not a valid measurement of layer 4 thalamocortical input. And again, my template for all of this is the 2007 Nature Neuroscience paper, and you can see just how perfectly these line up. The sink and the source in red and blue are just in the same spots. Now again, I don't quite have the x-axis scale bar that set. This is 0 to 100 milliseconds, this is 0 to 50 milliseconds, so I'm going to analyze this original data just showing you the first 50 milliseconds so that it's in alignment with this. And that's what I'm showing here. And here you can really see just how incredibly analogous this is. That you see the same patterns of activation, and that this sink is clearly at a depth of only 200 to 300 microns. And again, it's really rather tragic that the analysis in the lab for 7 years consisted of comparing layer 2-3 with itself. It's a huge missed opportunity. So just comparison of the data from figure 1B and the data from figure 1A. This is the figure 1B, it's a sink in layer 2-3, and this is a sink in layer 2. And here's comparison of the figure 1A and figure 1B. Again, the analyzed sink was just solidly in layer 2-3, which is at a depth of 200 to 300 microns. Okay, now I can get into more depth with figure 3A and 3B. And this is really, figure 3A is really an incredibly wild figure, just because these recordings are from the full depth of auditory cortex, and layer 1 is not on the y-axis. So here's the original figure. Again, no quantitative y-axis, no indication of what cortical depth these traces are recorded from, and no really account of why layer 1 is missing on the y-axis. Here's my analysis of the original data, showing that this sink is at a depth of only 100 microns. And here's lining up the anatomy with us, so you can see this is solidly in layer 1. If you recall earlier, I showed you that the full depth of auditory cortex is about 1100 microns. And they're putting layer 4 right here at a depth of 200 to 300 microns, which is layer 2-3. So it's not founded for them to say that this is layer 4, and especially since their measure of activity in layer 4 is not in here. Their measure of activity in layer 4 is right here. When they draw this line, they're saying that this is their measure for layer 4, which is at a depth of only 200 microns, which is layer 2. It's just incredibly shallow. And again, this is a failure to use Occam's razor. What's the simplest way to account for this data? The simplest explanation is that this is a layer 1 sink. That's a much more straightforward hypothesis than positing that layer 1 of cortex is above the surface of the brain. And why is layer 4 at a depth of only 200 microns? And where's layer 1? And you can do the same thing for figure 3b, where this is a very superficial sink at a depth of 100 microns, which is, this is a layer 1 sink. Here's another way of showing the same data from figure 3a. And if you read the methods section of the paper, it's very clear that in stating that the top recording site is visible above the brain. They say the top 1 or 2 recording sites of the multiprobe were above the pia and visible using a surgical microscope. Which begs the question, if these recordings were from the full depth of auditory cortex, which they would be if the top recording site is visible above the brain, then why is layer 1 not on the categorical y-axis? And why are they not forthcoming about the actual cortical depths these traces were recorded at? And again, this is the data from figure 1b. Now, what I have shown here is just a different way of analyzing the data from figure 3a, where I've increased the sensitivity on the CSD color plot so that I can pick up these smaller sinks. And when you increase the sensitivity, you can actually see that there is a sink down here and there is a sink down here. And in fact, the sink happens to be right in the middle layer. So this is really the early input into layer 4, which they missed completely and never analyzed. This is the activity up in layer 2, 3 with a layer 1 sink. And here's activity in layer 5. And of course, this is the canonical cortical microcircuit. You have activity starting layer 4, going up to layer 2, 3, going down to layer 5. And if you compare the original figure with the anatomy, you see that everything is mismatched. They put layer 6 where layer 5 should be, layer 5 where layer 4 should be, layer 4 where it's really layer 2, 3 at a depth of only 200 to 300 microns. They put layer 2, 3 where layer 1 is, and then they run out of space and just don't put layer 1 anywhere at all. It's really pretty wild. And I think I already showed you this figure, showing you that I don't understand how they can put layer 4 at a depth of only 200 microns in both figure 3a and 3b. Now I'm going to show you figure 5a, which is actually very similar to figure 1. So here's the original figure. The 5a, there's two parts of 5a. There's CF and non-CF. You don't really need to understand what CF and non-CF is. So most notably, notice that, again, these recordings are from the full depth of auditory cortex. Why is layer 1 above the surface of the brain? And what are these cortical depths that they're labeling as layer 4 and layer 2, 3? So here is a quantitative analysis of the original data where I have the actual cortical depths on the CSD traces here, and you can see the positioning of the multiprobe. And this is really a very superficial sink at a depth of only 200 microns. And when they draw this layer 4 line right here, they're saying that this is their measurement of layer 4 activity. Really, this very superficial sink in layer 2, 3 at a depth of only 200 to 300 microns, that's their measure of layer 4 thalamocortical input. Now I can do the same thing for the second part of figure A, which is this non-CF. And again, why is layer 1 above the surface of the brain? And this reveals that this is a sink at a depth of 200 to 300 microns. Now, this is a quantitative analysis of the original data from figure 5a, where I've brought everything to scale with this 2007 Nature Neuroscience paper figure, which I've been using as my template for the quantitative analysis. And you can see that this trace is at a depth of 200 microns, and you can see that the pattern of activity with this superficial sink is entirely analogous. You have this blue source up here, you have this red sink. And this is just simply a sink, a very superficial sink in layer 2. And same thing with 5a non-CF. Actually, in this one, you can see how entirely analogous this is. I mean, this is just the same pattern of activity, which is consistent with what I've been telling you, that the top recording site is above the surface of the brain and the second site is right at the surface of the brain. And that's clear here since you have the exact same pattern of activity. So now I'm going to talk a little bit about what happened to me in my lab as I tried to convince my lab to adopt a quantitative approach to analyzing and representing the data. So here is a figure of some of my own multiprobe recordings in the lab and how I wanted to interpret and present the data. I wanted to take a very quantitative approach to analyzing the data and just have a CSD color plot that had the actual cortical depths on the y-axis. And I wanted to make direct comparison of this plot with the data, with the anatomy, just put it side by side with the anatomy. That way, other scientists could look at the patterns and decide for themselves what it was that they saw in the data. And what I saw in the data was that here's an early sink in the middle layers at a depth of 500 microns in layer 4. And then there's later activity up in layer 2, 3, followed by activity in layer 5. And this is, of course, the canonical cortical microcircuit. Now, I was not allowed to present this in posters at SFN or at ARO or to publish any of this work or to really continue with my science in the lab because this was completely at odds with my advisor's framework. And my advisor's framework posited that this is a layer 6 axon collateral, this is layer 4 activity at a depth of only 200 microns, and that this is layer 2, 3 up here, but that's at a depth of 100 microns. So it's really pretty astounding that there's no quantitative y-axis here. It's just there's this assignment of what the layers are without any reference to the actual cortical depths and without any reference to the actual anatomy of auditory cortex. Now, on August 1st of 2011, I sent an email to the lab really making an attempt to try to convince them to take a quantitative approach to analyzing and representing the data. And my very, very first suggestion is that we should put depth values in microns on the y-axis. And my second suggestion is that we should put y-axis grid lines so that we're aware of the limited spatial resolution of our recordings, since we only have a sample every 100 microns. So those grid lines would look like this. I thought it would be a good idea to put these grid lines on here so that we really know what cortical depths these are at. We know that this is a sink at 500 microns in the middle layers. We know that this is at a depth of 200 microns, which is very superficial. Now, I sent this email on August 1st, 2011. 2 days later, on August 3rd, it was decided that I would no longer be a member of the lab. And my email was never responded to. No one ever responded to any of the questions or concerns that I had expressed. There was really no scientific discussion of anything. It was just decided that I would no longer be a member of the lab and that I had to find somewhere else to continue my graduate student education. And this is after I'd been in the lab for over 2 years. So my last day in the lab was August 8th, 2011. And I later found out, 2 months later, that this 2011 Journal of Neuroscience paper was submitted the day after my last day in the lab. Now, I had never seen these figures, the figures in the 2011 Journal of Neuroscience paper, until two months after I'd been out of the lab, when I saw it published on the Journal of Neuroscience website in October. And I was really shocked that they had put layer 1 above the surface of cortex. And in fact, even if you look at this figure right here from the 2012 paper, which just came out a few months ago, which came out after the 2011 Journal of Neuroscience paper, suddenly layer 1 is back over here again. Here's layer 1. I mean, the inconsistencies are pretty incredible. Now, I left the lab. I started all over in a second lab. And 9 months later, in March of 2012, I attended the Cold Spring Harbor Laboratory Neuronal Circuits meeting, and I presented a poster of my own work with multi-probe recordings, where I have a quantitative approach to analyzing the data and where I present the data right alongside the anatomy. So if you look at this figure here, I have my recordings, my LFP traces, my CST traces, my color plot, all alongside the anatomy. And this is really small, so I'm gonna magnify this so that you can see what I'm talking about. Here's what I presented at the Cold Spring Harbor Laboratory meeting. Here are some of my LFP traces. Here are my CSD traces. Here I have a color plot with the full depth of the activity in the full depth of auditory cortex. And I have this alongside the actual anatomy. Now, this is completely at odds with my lab's framework, which posits that this is a layer 6 axon collateral, and that layer 4 and layer 2, 3 are both in this very superficial sink. And I can show you how at odds it is with that if I just superimpose it right here. So my lab's framework posits that this is layer 6, right here in the middle layers, at what is really layer 4, and that this is layer 4 and 2, 3 up here at an incredibly shallow depth of 200 to 100 microns. I mean, this would be in layer 2. So what happened was I presented this poster at Cold Spring Harbor Laboratory, and this was during my spring break that I presented this poster. And when I returned to my now second lab, a week later, I was dismissed from the lab for presenting this poster. And it was actually characterized as graduate student misconduct. I didn't realize that presenting a poster at a scientific meeting qualified as graduate student misconduct, but apparently in my department it does. And again, here's just comparing the lab framework on the left with my own multi-probe recordings. So now I want to talk about some questions and concerns that I have, or that somebody watching this might have. So I'd anticipate that someone might say, okay, you've shown all this quantitative analysis using this 2007 Nature Neuroscience paper as your template for that quantitative analysis. And in that paper, the top recording site was above the surface of the brain, and the second site was at the surface of the brain. But what does the paper itself say, this 2011 Journal of Neuroscience paper, say about the positioning of the multiprobe? The paper in the methods section says the top 1 or 2 recording sites of the multiprobe are above the pia and visible using a surgical microscope. And everything that I've shown you has had 1 recording site visible above the surface, not 2. So let me try to illustrate how things might look differently if it was 2 recording sites visible above the surface instead of 1 recording site visible. So here's the original figure 1. Again, categorical y-axis, no cortical depths on here. So I'm going to add my analysis, the original data right here. I'm going to add a quantitative y-axis. And this is what everything looks like if there's only 1 recording site visible above the surface of the brain. And you can see that these analyzed traces that they have are depths of only 200 to 300 microns, which again is solidly in layer 2-3. Now, again, this is with the top recording site visible above the surface. What would this look like if the top 2 recording sites are visible above the surface? Well, if you did that, then this sync would actually be at an even more superficial depth. This would be 100 and 200 microns they'd be comparing. So really, if by giving them 1 recording site visible above, I'm really giving them the benefit of the doubt and analyzing the data in a way which is consistent with what's described in the methods section. So again, with 1 recording site visible above, it's 200 to 300 microns. With 2 recording sites visible above, it's 100 to 200 microns. So this doesn't help their case. The sync actually gets even shallower. Now I can do the same thing with figure 3a. This is with 1 recording site visible above. Here's with 2 recording sites visible above, and the sync just gets even more superficial, actually to an absurd degree. And you can also reference an email that I have reproduced on the site, but with the original names removed, where the third author is really very, very clear about the positioning of the multiprobe. In the email, he mentions the visible recording site above the brain, and then goes on to mention that, of course, everyone should make sure that their second recording site, the first CSD site, is as close to the surface as possible. So that's entirely consistent with what I've shown you thus far. So the top recording site is above the surface of cortex. The second recording site is at the surface of cortex. And that's clearly described in this email from the third author, which is again on the site. And if you want to see the original email, you can just write to me and I'll forward you the original email. And in that same email, the third author goes on to discuss the fact that the sync is at a very superficial depth of 260 microns, which places it in upper layer 3. So it's not as if the authors were ignorant of what cortical depths they had recorded at. They were entirely aware of the fact that this was a very superficial sync at a depth of 260 microns, and that this was upper layer 3. So figures 1, 3, and 5, all of them do not have a quantitative y-axis. There is no cortical depth in microns. And all 3 figures boldly posit that layer 1 of cortex is above the surface of the brain. Again, in figure 3a, they put layer 4 at a depth of only 200 microns, which is layer 2, not layer 4. And we have to ask why they placed layer 1 above the surface of cortex when the recordings were from the full depth of auditory cortex. We saw the same thing in figure 3b. In figure 3a, we saw that just as in the 2007 Nature Neuroscience paper, the analyzed sinks were at depths of 200 to 300 microns, and their analysis consisted of comparing activity recorded in layer 2.3 with activity recorded in layer 2.3. So their analysis consisted of comparing layer 2.3 with itself, which is rather embarrassing. So you can see perhaps why it is that the figure avoids a quantitative approach to representing the data. It just seems engineered to obfuscate the fact that these two traces were recorded at very superficial depths in layer 2.3. And in figure 1b, we saw the same thing, that this is a very superficial sink in layer 2.3, and that their measure of layer 4 really isn't supported by a quantitative approach to the data, because what they're calling layer 4 is a trace at a depth of only 300 microns and is a sample of a layer 2.3 sink. And this has been the mantra throughout this talk, is the fact that a depth of 200 to 300 microns is layer 2.3. So when they're comparing these traces recorded at depths of 200 to 300 microns, all they're doing is comparing layer 2.3 with itself. Here's the data from figure 1a, where I've brought everything to scale. Here's the data from figure 1b, which is entirely analogous. You can see that just everything is really lined up here in exact the same. You can tell that this experiment was done the exact same way as was done here. Here's the data from figure 5a CF and from 5a non-CF. Again, you can see this is a sink at a depth of 200 microns in layer 2. This is a sink at a depth of 200 to 300 microns in layer 2.3. And finally, figure 5a, when I analyze the original data here, it shows that this sink is at a depth of only 200 microns, a very superficial sink. And of course, we have to ask the question, why is layer 4 at a depth of 300 microns? Why is layer 1 above the surface of cortex? And we see the same thing in the second part of figure 5a. This sink is at a depth of 200 to 300 microns. Layer 4 is at a depth of 300 microns, which is inaccurate because layer 4 is actually down here from 400 to 500 microns. And we have to ask, why is layer 1 above the surface of the brain? And again, the method section is very clear that the top 1 or 2 recording sites of the multiprobe were above the pia and visible using a surgical microscope. So the simplest way to interpret this data is to say that this is a layer 1 sink, especially given that it's recorded at a depth of 100 microns. That's a much more straightforward interpretation than positing that layer 1 of cortex is above the surface of the brain, or that layer 4 is at a depth of only 200 microns, which is absolutely not supported by the anatomy. And you can see here in this figure that these laminar boundaries are entirely unfounded. They put white matter at where layer 6 really should be, layer 6 where layer 5 should be, layer 5 where layer 4 should be, layer 4 where layer 2.3 is, layer 2.3 where layer 1 is, and then they just run out of space and don't even put layer 1 on their categorical y-axis. Why is layer 1 not in the figure? So it's a layer 1 sink, but the authors leave layer 1 off their categorical y-axis and present the data as if they were analyzing layer 4 activity at a depth of only 200 microns, even though the sink is at a depth of 100 microns. So their conclusions are absolutely not supported when you take a quantitative approach to looking at what cortical depths these traces were actually recorded from. And we really have to ask the question whether it's really valid to posit that layer 1 of cortex is above the surface of the brain. Is this really a valid, scientifically informed perspective? And again, the experimental paradigm, these multi-probe recordings from auditory cortex, I think the most disturbing thing in all of this is the fact that it seems that the authors intentionally avoided a quantitative approach to representing their data. They knew that the analyzed sink was at a depth of 260 microns. They knew that they were only comparing 2 traces in layer 2.3. And it just seems to me that perhaps they were embarrassed by this, by the fact that these traces were at a depth of 200 to 300 microns, and that they really had just compared layer 2.3 with layer 2.3 for years upon years, and were just bashful and didn't want to admit that, and thus left the cortical depths off the quantitative y-axis. And I suppose maybe I'll close or head towards the exit with showing the analysis of figure 1a and 1b again, where you can really see just, I guess I want to show these just to show that my quantitative analysis of the original data really is very consistent with this template that I've taken from the 2007 paper, because you see the patterns of activity are at the same depths. For 5a non-CF especially, and for figure 1b. And these, you can see that these are just really superficial sinks, and that the analysis in the lab was really limited to just analyzing this layer 2-3 sink and comparing it with itself, which is a really tragic and really a missed opportunity, because they never analyzed this early middle layer sink. They just completely ignored it and called it a layer 6 axon collateral and just never analyzed it. They actually never compared the sink here with this sink up here. And again, you can clearly see that this sort of the trend in the lab has been to avoid a quantitative approach to analyzing and representing the data. And instead they have these figures like this one from the 2012 paper, where there's no quantitative y-axis, and they just label these sinks with these layers, and they put layer 4 and layer 2-3 on the superficial sink, which is a very superficial sink at a depth of 200-300 microns, and they put layer 6 on this middle layer sink. So I just think that the framework is unfounded. And another disturbing thing is the fact that from the same 2012 Journal of Neurophysiology paper, they make it clear that they're entirely aware of the cortical depths. They even mentioned that upper layer 4 is a depth of 400 microns, which is correct actually. And they mentioned that layer 2-3 is at a depth of 200 to 300 microns. So they know that this is layer 2-3. They're entirely aware of that. And yet in this 2011 Journal of Neuroscience paper, they don't communicate that to the readers, editors, or reviewers. And again, if you look at this email, which I've reproduced on the site and which I can forward to you if you'd like to see it, it's very clear that they're entirely aware of the superficial depths of the analyzed sink. And the email is also very clear about the positioning of the multi-probe, which is entirely consistent with all the analysis that I've shown you thus far. So figures 1, 3, and 5, there's no quantitative y-axis. Astoundingly, layer 1 of cortex is placed above the surface of the brain. And the analysis throughout consisted of comparing a very superficial layer 2-3 sink with itself.